Hi, Priya Ragini. We have not started yet. We are going to be starting in 10 more minutes. So thank you for joining today. So we are starting in 10 minutes from now at 9 a.m. CST, which is 7:30 p.m. Indian Standard Time. Thank you for your patience and for your time. And as you are joining in today, please keep your microphones muted and your videos turned off. This session is right now live streaming on YouTube if you would want to watch there. And also we will be recording this session. So thank you again. And if you have any questions, the chat box is open. So please post all of your questions there. I want to start by saying that I am Deepsha Biswas and I'm the community manager here. You can also introduce yourself in the chat box. So where is everybody from? Can we have some answers in the chat? Okay, hello Sameet from Pakistan. Hi Prashant, thank you for joining today. Hello Tanmoy, how are you doing today? Hello Bibek. Thank you. Hi Fiza. Thank you all for joining today's session. So can you tell me what is your current interest in research? You can again put that in the chat box so that we can get to know each other through this interactive session. What are you more interested in studying? Is it oncology? Is it virology? You can put any of these down in the chat box. Or if you're just starting and to explore bioinformatics. Okay, Bibek, great. Okay, thank you, Prashant. Very interesting. Hi, Bishwa. Thank you for joining today. Thank you. So let us wait so that other people can join as well, because the time is still at 9 a.m. CST. So we are going to be waiting for everyone to come in the session. If you have any questions for us again, then you can post them in the chat box. Thank you so much, Ilmark, for joining today. We are glad to have you with us. Hi, CA. Welcome to today's session. Okay, hi, Christian. Hello, Rahul. So all of our sessions that you will attend, whether it's a free webinar or any of our programs, they're all interactive. Yes, yes, Christian. All previous webinars are there on our YouTube channel. I will be sharing the link shortly and also email everybody the links to this session and to any other previous sessions that you may be interested in. So we love to see that everybody is very open and communicating with us right now. So we would want you to participate in this whole session. Hi, Winfred. Prashant, about the programming part, we will be having some demo and we would hope that you can join us for this session. But as this is just a 1 hour session, maybe we are not able to do it at length. So we will be discussing more about that. Hello, Suraj. Good morning to you too. Good evening then. So yeah, we are just 6 minutes away. Yes, thank you so much, Ilya, for sharing the resources. Hello, Soham. So we are going to be discussing and showing you how to use these resources, but you can still start exploring these resources if you are available and watching this from your desktop. Then you can go to edu.t-bio.info and see and register yourself as well. Yes, and you can register for free already on edutbioinfo and code.omicslogic.com. And in today's session, the mentors are going to be giving you an overview of these platforms and how you can learn about NGS data analysis through these platforms. Hello, Dilip Sundar, and good evening, Fmind. Great to have all of you here. Yes, thank you, Ilya, for sharing this course link. So this is a free course that is available. You can right now go ahead and sign up for free and start taking this course, which is the Edu Bioinfo course right now on the platform. So this is a free course which is available uh, for everyone, and anybody joining for the first time can start with this course. Suraj, we will be getting there soon, as the session mentors would be talking about these today. Thank you for your patience. We just have a few more minutes to go. Thank you so much for waiting today. Okay, wonderful, Samit. That is great to know. Yes, so Samit, if you take this introduction to bioinformatics course, you will also get to know about the biological terms that are explained within this course. And you can start your bioinformatics learning from there. Because in our experience as well, there have been students from computer sciences background or any other backgrounds who have also been able to understand this by starting with these introductory and preliminary courses. So you can start with this free course and explore. And again, there is a glossary as well, which is available. So we will be showing you a demonstration of this very soon, as soon as the program starts. So let us wait and see. Yes, sure. I will send you the course link again. This is the course link again, which is called Introduction to Bioinformatics. Yeah, thank you. We do have a WhatsApp group. I can share that link soon with you. For the code playground, you just need to log in via the same account. So yeah, you should be good. And please use the same email across all the platforms, because the email is the identity that we use to identify you. Okay, wonderful. So I think we are ready to start now. Hello, everyone, and welcome to the online session on Omics Logic Introduction to Bioinformatics. This session will again be recorded, and it's also being streamed live on YouTube. So thank you for joining today. Okay. So welcome to today's session. This program is an introduction to the field of bioinformatics, which is an intersection of informatics and biology. Omics Logic is a program for bioinformatics training that was developed in collaboration between Pine Biotech and the Tauber Bioinformatics Research Center. What we will discuss today and throughout this program is a result of hard work by an amazing team led by the company co-founders, Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Alfred Tauber comes from a medical background and has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center at University of Haifa and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team includes experts in bioinformatics, computer science training, and some of our team will be presenting today as well. In the upcoming Omics Logic program, you will have the opportunity to interact and collaborate with an international team of experts. Let me introduce you to the mentors and program speakers. Ilya Brodsky is our CEO and co-founder at Pine Biotech. In his current role, he has extensive experience working together with industry and academic partners that are applying multi-omics integration and in bioinformatics to a variety of biotechnology and clinical applications. Julia Panov is a bioinformatician and has developed many of the resources we use for the Omics Logic training. Dr. Harpreet Kaur is our Omics Logic trainer and an expert bioinformatician who will guide the hands-on sessions and assist participants with projects and analysis during the program. Her area of specialization is cancer genomics and machine learning. Dr. Mohit Mazumdar is our Omics Logic project mentor and an expert in big data and computational biology. Now, I would like to invite Dr. Mohit Mazumdar to take over today's session.
Thank you, Deeksha. And hello, everyone. Let me share my screen. So my name is Mohit Mazumdar, as Deeksha said. I'm one of the global representatives of Pine Biotech. In my current role, I'm collaborating and working with academia and industry to provide support for bioinformatics education and research. I have 12 years of experience working and collaborating with pharma healthcare to solve high priority scientific questions related to biomedical data analysis and bioinformatics capacity building. So in this program, we will discuss the changing landscape of data-driven research in life sciences and the opportunities that come with the widely accessible high throughput technologies. Various data types and these emerging technologies have shown a tremendous impact on healthcare, agriculture, environmental sciences, public health, and more. So biology is rapidly acquiring the characteristics of data science. Billions of data points on genes, proteins, and other molecules are compiled in these large files and have been systematically studied. This influx of data, associated methods, and data-driven research makes bioinformatics for life sciences as prominent as the emergence of computers for the tech industry. Analyzing this kind of data will lead to more knowledge and understanding about living organisms. And this introductory course is designed for an overview of bioinformatics for students, biologists, and clinicians. We will have a first session on 13th October 2020. Sorry. And we'll cover some of the important technologies as its and its application in today's world. So let's talk more about this transformation of biology to data-driven biology. So more and more biologists and clinicians are now expected to understand and handle their own data, and uh, and the expectation is to understand their own data to publish and succeed in both basic and translational research. In fact, biology itself is rapidly acquiring these characteristics based on the data that we are generating from different experiments. So this program will give you an overview of bioinformatics for for beginners and biologists who are working on the lab, and also clinicians. So in this program, we will combine online interactive sessions, self-guided study material, and practical assignments for an immersive experience that has proven to be effective in our well-known Omics Logic research training program. So the coursework will provide introductory materials that have been taken by thousands of people around the world and have been rated very highly. So online courses cover important terminologies and provide a perspective from experts on this exciting field. Each courses have quizzes that allows you to see how much you have learned, and all this activity is measured using our point system that lets us identify any technical issues and resolve roadblocks that are so common in bioinformatics. So in at the end of this program, each student will receive a personal certification of either participation or excellence based on their performance, and and you will experience our project-based approach to learn bioinformatics, where we take data sets from high-impact publications and use them for practical exercises. These projects include topics such as oncology, neuroscience, agriculture, and others. So as a result, anyone starting on this journey in bioinformatics can start developing a project of their own. So by seeing how experts analyze their data and achieve research results, you can start developing a research question of your own, identify the appropriate type of data, and plan your analysis. So this introductory course leads into multiple courses and projects that serves as a springboard, into this world of bioinformatics, big data bioinformatics. So to access these resources, we will use the educational platform. So why don't we go to the educational platform and create a login first to learn about the resources. So many of you who have access and who have been working with us through various programs must have the login credentials. But to those who are joining for the first time, why don't we do that? Why don't we go and do it ourselves? So Deepsha, if you have already shared the...
Yes, the link is in the chat box.
Thank you. Okay, so this is the link. This is the place. If you go to edu-t-bio.info, and if you are already here, please let me know by writing yes in the chat or there in the chat. Thank you. Thank you, everyone who have already replied. I see Edwin says yes, Suraj, and many more. Where were we? Thank you. So now let's try to log in. So I already have a login, but I can easily make a login, or I can log in using any of these accounts. So why don't you go ahead and log in? And meanwhile, I will try to log in as well, because my credentials are already stored with my Gmail account. It has been associated. So just let me log in and let me know if you guys are there, like once you guys are there. So if you have already logged in, then we will look into the resources, and we will do this hands-on exercises later on. So it is kind of really important to do this part. If you want to practice a hands-on session, uh, that will be that we will be doing. Sorry, I'm just not able to see. So maybe because I'm using a incognito, let me do it in a new tab. So if you guys are there, logged in, let me know. Anyways, so to look into the site, we will be exploring different sections in this platform, and where we have courses and projects. So Bipsha, if you can show this part, I think I'm having some trouble with the cache. So can you do that? Help me.
Yeah, sure. I will show
I'll
everybody
just
how.
stop the sharing.
Yeah, sure. I hope you all can see my screen for those of you who are having a problem. Okay, so this is how I hope you're able to see my screen. So it's going to be, yeah. So you see, so I'm logged in already here. So if I logged out, you will be finding there's a register or a login option right on this side. So what you do is that you go to the register option if you're logging in for the very first time, and you use any of the options that are available to you to log in. So once you are logged in, you will see that you come across all of these different resources. So how many of you can see all of these resources? Can you see all of these resources? So a while ago, we were talking about introduction to bioinformatics, right? So yes, okay, great, Prashant. So can you all come to introduction to bioinformatics? Okay, perfect. So we are here on Introduction to Bioinformatics. So this again is a free course. So you see, I had already taken this course. So it shows me that I can retake this course again. So if you go down, you can read in the overview about the course, what it is about. And you read through here and you start by taking the course right here. So there is an intro survey, which all of you should take. The intro survey helps us learn more about you. And how is that useful? That is useful because we can then understand, okay, you are looking for this particular type of training, and we can point you to the right direction or help you out in your research. So that is why there is this intro survey. So you can start by filling up the form here. Let me also just post this in the link right now, the chat. Okay. So it might take a bit more time to load. So this is the free course. Please take it. And there are 3 other free courses. So if you just go down here, you can see there is a course collection. No, you do not need to do it again if you already finished the course. Because once you finish the course, you will of course have a certificate that is added to your profile. And I will show you the certificate later. So you see there is this free course and the free survey. So once you start, these are what we called asynchronous courses, means courses that you can do on your own, and they are provided as part of the training. So you will see that these courses come with a video, there is text, there are images to explain to you. So as I remember that if you are starting with big data bioinformatics or just starting with biology and you are not familiar with a lot of the concepts, you can see here is this highlighted. So if you just hover over this term, you will be able to see what you means by big data or what you mean by proteomics, right? So you can learn more about these terms as well. You can explore the different resources. At each of these sections, there is also quizzes and a demo. So if you go down, you will be able to find the quizzes. You see there are 10 quizzes right here. So why? Because I finished it, you will see each of these section is tick marked. So please explore these courses. And now I would like to pass it back to Dr. Mohit. Dr. Mohit, would you
Yeah,
like to
thank you,
please?
Biksha. Yes, please. So that was the demonstration of the first platform. And if you guys have already logged in, so kindly start exploring. And once you log in, you will have a profile. So kindly update that profile so that we can learn about you and we can reach out to you if you need any help with any of the bioinformatics resources that we are going to talk to you about today. So going back to the presentation,
Dr. Mohit, you're muted. Can you please unmute yourself?
okay, sorry about that. Yeah, I was saying stuff. So yeah, thank you, Biksha, and thank you for the demonstration. So that was the first platform, the educational platform, where you will find the courses and the projects. And now moving back to the presentation. So the next platform, a very interesting platform, is the T-Bioinfo platform. So for the duration of the program, we will be using the T-Bioinfo platform to process big data sets. This platform is built for multi-omics integration and analysis of structured data with advanced methods such as machine learning and mathematical modeling. And we process almost all kind of NGS data using the platform. So this is the interface of the platform. And if I go here, if I show you this, there are different sections. So you can see sections by data types, NGS data, mass spec data, structural data, and others. And then you will find demo pipeline. So run analysis and play around. You can play around with demo pipeline. So we are going to show you and demonstrate you and do a hands-on session with you today on one of these pipelines. So please have patience and we'll be sharing more details. And the third part which we will be covering in this program is to be able to learn about visualization, statistical analysis, and machine learning. So you can also find your pipelines once you are logged in and you are a user and you are a participant. You will be able to see your pipelines and the analysis that you have already done. And you will have a login information, obviously, and account information to be able to log in into this platform and start working on the data. So for today's session, we have this special hands-on session that we wanted to do with all of you. If you have the access to a laptop, that is preferable. So the prerequisite is to go to this site. So let's just go to this site. And then we will use this username and password. All of you can use this username and password to log in into this server, T-Bioinfo server, where we will do some practical analysis on real data sets for further learning and this hands-on experience. So let's just go there, server.t-bio.info. So this is the server platform, and let's just try and log in. So all of you, I think Deepsha is sharing
Yes, it is shared in
the
the
details.
chat. Yes.
Perfect. So I am going to copy it from the chat like everyone else and try to log in using this credentials. So let's do that. So
Please let us know once you are there. How many of you have been able to go to the server TBio platform?
And
Put down.
log in.
Great,
So,
Shreya.
so you can log in using 123 edu test by writing this. So I just copied it and I just pasted it on the website, and I'll just log in. I'll try logging in. So let's see what happens. Voila, I'm in. That's good. So if you guys are in, let us know that you have the access. You have, yeah. So now you can explore for yourself the different sections that are there. And today for this session, we will be demonstrating one of the pipelines that we will be covering in much more detail in the full program, Introduction to Bioinformatics. But I think I'm glad that if you guys have been able to log in. So let's look at, I mean, have you been? Yeah. Sangeeta, yeah, thank you. Shreya, Sonia, great. Irmak, yes. Edwin is in, great. There's so the password is 123 edu test 123 edu test, and you can log in like from here. Let me do it again for those who are not being able to do it. Just like go here in the site server and then try logging in here, and then info at pine biotech.com, and password is 123 edu test 123 edu test, and enter. And I mean, okay, so please explore this, and I'll go back to the presentation, and we will use this after some time. So I think it would be great if you guys can do it. And Sabila, if you have not been able to, then take your time. And after some time, we will have after some time we will have that demonstration, which will be great if you guys, or if you can all do it along with us. So going back to the slide, the presentation. So this is the second platform, the analytical platform. So the third platform is the coding platform. So dealing with data efficiently to process, analyze, visualize, and annotate will ultimately require some coding. So even if the code launches other scripts developed by a more experienced programmer, yet you would need and require some coding understanding to be able to execute that. So the Omics Project Code Platform is an easy to use platform that helps non-programmers to learn the logic of coding. In practical coding assignments, these practical coding assignments will help you understand the R and Python code elements that will be part of any bioinformatics project that you end up working on in the future. So these exercises are designed to explain the code and help you adapt it to a project of your own with clean and efficient code practice. Today, we will discuss the analytical and the coding platform in the context of this program when we review the session in some time. So, in summary, I mean, this course provides a broad overview, but is structured to offer significant depth even for the experienced users or people who have experience in bioinformatics. So after completing this short introduction, I guess you will be inspired to continue learning about this exciting field of bioinformatics. So again, to know you more, so I'll take a pause here and see if we have any specific questions. So Bisha, please let me know if we have any questions that I might need to address.
Yes, so there was one question from Shushmita. She had asked, Could you say about how to characterize novel peptides using mascot analysis or any other in your talk kindly?
So that is a quite that is sort of a kind of a specialized question, and thanks for asking us. And if you can let us know your email address, if you let us know, or let Beeksha know your email address, we can help you get an answer to that question. But it will be not possible to do cover it for today's session. However, uh, okay, is there any other question, Beeksha?
Okay. Yeah. Shushmita says thank you. So Shushmita, you can reach out to us at marketing@pine.bio. And also she has left her email. So okay.
Okay,
Okay.
wonderful.
So
Thank you,
there's a
Sushmita.
question from Farheen. What if students from conventional biology do we need to learn R?
So you don't need to learn R, and that's the benefit of using the platform. So if you are interested in learning about the analysis, if you have data and you want to learn the analysis and you want to reach to a significant result where you can talk about that result, maybe you want to publish that result or whatever analysis or research question you're trying to address. So it's not mandatory to learn R or Python because we have these platforms that are built for biologists and clinicians to be able to work on their own data without any coding experience. But we also offer this coding experience through this intuitive platform, which we will be demonstrating in a while, where you will see that it's not so difficult after all to learn coding, and you can give it a try. And if you're not comfortable, there are other ways that you can work with that data and get useful results. So I think it's just for those who are interested in doing more in bioinformatics, doing their own research, or trying to learn new techniques. So that adds on.
Yeah.
So yeah.
Dr. Mohit, I will just address a few questions here. For Brian, just to clarify, please talk more about how to install Arc Studio. I will share with you a blog right now. We have listed it out how to download R and Python. So we will be sharing that with you. And we'll also be demonstrating the coding platform today. So I know everybody right now has a question about R and Python. So we will be addressing this in just a few more minutes. Okay. Okay. So Webaby here is asking a question. She says that JavaScript is the most popular language followed by Python in general. Is it commonly
So
used in bioinformatics as well?
and thank you, Ilya, for answering the question. So we already have a team on chat. So I think everyone is contributing, and uh, so I can move on with the presentation, and we will answer these questions when we get time. So we will also be emailing you if there are like a list of questions that are interesting. So thank you. So uh, what I wanted to say is that I've been talking for a while, and I realized that we are a large group and we don't know each other so well. So and we haven't had a chance to introduce each other. Uh, so let me ask you a question, if you don't mind. So you can answer it in the chat box. So A, B, C, D, E. So if you're an undergraduate, if you're doing post graduation, or you're done with post graduation, if you're a PhD student, or you're a postdoc or a faculty, please let us know, and that will help us to understand you better and to give you right resources which we have prepared. So another aspect is to know about, maybe if you can tell, a lot other than this, if you feel, you please feel free to write more if you have any specific interest, like working on neuroscience, or maybe more specific if you're working or if you're interested in Alzheimer's, or some aspects of cancer research, or precision oncology, or infectious diseases. So many students that I have interacted with were keen to learn data science for bioinformatics, for example. So we'll be really glad to know, if you can share something about yourself and your interest, and we'll be able to see this later and discuss and talk to you more through email and maybe interact with you on some in our program. So thank you so much for those who are sharing.
If I may add a couple of things here, Mohit.
Sure.
So
Yeah.
when you're answering these questions, it would be really nice for us to have also your contact information. So the way that we are structuring these free webinars is when you log in to the different platforms that Mohit described, the educational portal and the coding platform, you can create a profile. And if that profile has your full name and your email address consistent with what you are using in this session as well, we will analyze all of the data from this webinar, and we will also take a look at your profile to make sure that we can suggest the appropriate steps for you to continue your journey in bioinformatics.
And also to add to this, Ilya, if you could also answer Prashant's question, how is T-Bioinfo different from Galaxy?
That's an excellent question. The Galaxy platform is an excellent solution for those learning about bioinformatics using a visual interface. However, it also requires many input and output configurations that you only get to know as you become more of an expert in bioinformatics. In addition to that, Galaxy platforms are offered as a cloud version and as an installed version that you have to install on your computer. As you install it on your computer, you will quickly realize how limited your resources are to process big data sets. The T-Bioinfo platform is completely cloud-based, so you never run into the issue of using your own resources. And the interface is much, much more intuitive than Galaxy, if you will be able to see in today's session. Also importantly, every step of the way is not focused on the technical component, it's focused on the logic of analysis. So we'll see today how when you build the pipeline, you will be able to actually learn about what each step does, find additional publications on why we're using this method, and be able to compare multiple pipelines easily. So I'm hoping that in the demo part of this session, you'll be able to see what the differences are if you are already familiar with Galaxy.
Thank you, Ilya, and thank you everyone for participating. So talking about data, big data. So bioinformatics is now playing a key role, as I have been talking about, and in all healthcare industry and pharma industry especially. The pharma industry is now considering data science and bioinformatics to be an important part of their portfolio. So this data growth that we have seen during all this time, and you can see from this plot also. So yeah, an exponential growth. So these resources and data tells us a clear story that that as a scientist or researchers or student, there is a lot of opportunity that you can take using this. So in this program, we will be learning about the bioinformatics aspects and different data science aspects of our analysis. So before we move into the next part of this session, I would like to invite Dr. Harpreet Kaur to talk about her experience and discuss the program in much more detail. Thank you.
Thank you, Dr. Mohit. So let me share my screen. Hello everyone, my name is Harpreet Kaur. I have a PhD in bioinformatics. My research is on biomarker discovery for diagnosis and prognosis of liver cancer using bioinformatics. Now let me take you to the curriculum of this upcoming program. This program is designed to highlight the role of big omics data that is shaping modern biomedical innovation, where molecular data is used for precision diagnostics, big data integration, machine learning algorithms that are transforming personalization of a treatment selection. And there are various innovative algorithms that allow for unprecedented capabilities with drug discovery and design of large molecules and drug repurposing. During the upcoming program, in the 6 interactive sessions, you will get to see the broad applications of next generation sequencing, biostats, and tools for big data analysis. In practical assignments, we will be learning about the logics of the bioinformatics and will see how data science methods can be used to study biology. So in conclusion, you will also be able to plan how to take what you have learned and apply it to an independent research process using various tools and methods that we will cover during this program. As Dr. Mohit mentioned, in the the first session, we will learn about resources in much more detail for the big data. So once we learn about these resources, then we can obtain data from these resources and, and we will learn about data analysis using various platforms. So when we listen about the term genomics, what is the first thing that comes to our mind? So can you people tell me, or just write it down in the chat box? Okay. So for me, whenever I listen to this genomics term, I think about the genes, DNA, and maybe ATGC codes. So in this second session, we will learn about shotgun sequencing reads, mutations, and need for the structures data and associated bioinformatics methods. We will also understand about processing of data for germline and somatic mutations. And here we will perform various analysis that include mapping, detection of the variants, and we will also understand about the interpretation of the significant and insignificant mutations. To learn more, let's take a look at the visualization of the genomic data pipeline. So now let me go to the platform and let's see how we can visualize the alignment of genomic reads on a reference genome.
Yeah, so I will request everyone to let's go to the platform and let's do what Dr. Harpreet is doing all together.
So everyone, are you there? So just let me know so that I can understand that you are with me. So if you are there, okay. So yeah, now we can start. So this is a demo pipeline. So we have already provided the data. So you need not to upload any data here. So we have already uploaded this data. In this pipeline, we are.
If I may interrupt, Dr. Preet, if we can go back from the beginning and just like from the server, show us where this demo pipeline is.
Okay. Okay.
And
so yeah.
Yeah, and other people are trying to log in. So
okay.
Edwin, what problem? Edwin says he cannot log in. So Edwin, please try refreshing your browser and try again. And if any one of you are not being able to, log in, please look at the analysis that Dr. Harpreet is showing, and you can repeat it later as well.
Yeah. So if you're able to log in this server, so you will find this kind of piece here. On this interface, you will find an option of area of analysis. So you can click on this option. So you will get this interface. So under this interface, so under the NGS data section, here under the transcriptomics, we will find a first pipeline that is RNA-seq or chip parallel analysis of NGS and microarray data. So under here, we will find a drop-down option. So if you click on this first pipeline, that is demo, that is using RNA-seq cell line project. So here we have already are using one project that we have considered a cell line. So details of that project we will talk during the complete program. So now let's click on this demo RNA-seq pipeline. So every one of you able to find this option? Just let me know and the chat. Just post yes. So if every one of you with me, so clicking on a
wait, I think there's some issue. Ilya, I mean, people are not being able to log in again, I guess.
Yeah, it's. I will just add something. If you get the issue of invalid email or password, then you should check that there is no space before or after username or password. I think there's a lot of people connecting, so there could be an issue with that.
Anyways, if there is a lot of traffic, then what we can do is that we will be emailing you this entire video presentation.
I will share
Yeah,
a couple more logins so that people can use them simultaneously. I don't think we were expecting 100 people to join. So let me, you can go on with just showing this, and then we'll repeat it again. I'll just share some more logins.
Perfect. Dr. Harpreet, if you can go on with the presentation.
okay. So okay. So let me just should I demo here, Dr. Mohit, or I will demo it later?
Yes.
Okay.
Yes.
Okay.
Please.
Okay. So once we click on, you can even run this pipeline later on when you have complete access. So just let me show how we can analyze sequence alignment on a reference genome, how we can visualize that. So once we click on a start option, you will find an option. There are the different input files. So as this is a demo pipeline, so we have already uploaded our data sets. So here we have already provided the given samples as well as the reference genome. So once we click on this side, you will find various options for that are getting highlighted. So though there is a next option under the pre-processing, so under this you will find a different option. So I'm just as we just want to visualize here, so I'm just skipping this particular step. Whenever we are performing any analysis, then we can perform pre-processing. So I'm as for the visualization purpose, I can skip this step. So then we will go to the next step, that is the mapping of the genome. So here different algorithms are available that will align your raw sequence reads on a reference genome. So there are different algorithms like top hat 2 and Botai 2E. So I am here going for a Botai 2TG. So what it does, it will align your sequence reads on a reference genome. So then the next option that is getting highlighted, that is visualization. So once we click on a visualization, so it will take you to the server where you can visualize. So there are a number of the servers are available that are freely accessible, like JBrowse, IGBI, then UCSC Genome Browser. So with different browsers, you can visualize the alignment of the sequence reads on a reference genome. So here, the server that we are using, that is JBrowse. So even on clicking on every single step, you will find the details of each of these steps. So once we get this, after clicking on the visualization, then the next option that is getting highlighted, that is end. So if you click on this, it will complete your pipeline. So once you complete your pipeline, you will find various files under this download option. Beside, you will find one another option for the visualization. So we can click on this visualization to visualize our genome. So like here, so it will take to us here. So here we can observe this is a reference. This is a JBrowse piece where this is a reference genome. So these that is red and blue color are the reads that are getting aligned on a reference genome. So apart from that, as there are the if you look at the downside, you will find there are the different peaks. So these peaks are actually indicating the coverage. So higher the higher the height of this particular peak, higher the coverage of that particular sequenced region. So like here we can see there are large number of the reads that are getting to align to this particular genomic region. So that's why this peak is very this peak has higher height. So in comparison to here, if we look, there are only the few reads that are getting aligned. So you will find there is either a no peak or a very small peak. So this is how these you will find that how your raw sequencing reads are getting aligned to the reference genome. The what is the importance of these peaks is that uh, um, better, I mean, a higher the peak, it indicated there is a better coverage, high coverage. So high coverage means there is high confidence of that particular region that where your sequence is getting aligned. So if you click on these particular reads, so you will find details regarding this particular read. Like here you can find what is the position of this particular read on a genome sequence. Then you will find the quality of each nucleotide on the of your read with the quality score. This side you can also find the length of that particular read, and this is how you can find complete details regarding the sequence alignment on a reference genome. So I hope every one of you able to get this point. So now let's move back to. Okay, so you have some questions, Anshu?
Okay, so before you continue, there is another account that you can use.
Yes, I have shared it with a few people, Ilya, on chat
Okay.
to just see that if they can log in using that.
Okay, thank you.
Okay. Should I move ahead or should I give again a demonstration?
It would be great if we can do another demonstration.
Okay, that's okay.
Yeah, and so I see that a few people are having questions about how to actually get here. So maybe you can start off from areas of analysis.
Yeah.
And maybe those of you that are asking questions about what kind of input data this is, we're not using a full pipeline builder, so you don't need to deal with any kind of inputs. So Harpreet, could you please make
Okay.
sure that people see where the
Okay.
demo pipelines are?
Okay, so every one of you are on this particular interface. Just let me know that. So if you are at this interface, you will find here various options like NGS data, mass spec, structure, data integration. So under the NGS data, specifically under the transcriptomics, you will find a first pipeline, that is RNA-seq parallel analysis of NGS and microarray data. So here you will find a drop down. So if you click on this particular drop down, you will find here three demo pipelines. So the pipeline that I was demonstrating here is the first pipeline, that is RNA-seq demos using cell line project. So every one of you able to get this point?
And was everyone able to log in using the new login ID? I've shared it with a few people. Okay. Dr. Sunil, were you able to log in using the new ID that I shared just now? Okay. Let me share it again for everyone so that you can follow through. So this is the ID. Yeah. Can everybody try again?
Yes, Dr. Harpreet, if you can quickly, we are running out of time and we have still
Okay,
slides to cover.
so if every one of you at this space, you will under the NGS data, there is a transcriptomics subsection. So under the transcripts of subsection, you will find first pipeline, that is RNA-seq pipeline. So under that, when you will find a drop down, and under the drop down, you will find 3 demo pipelines. So we are going with the first pipeline, that is demo of RNA-seq using cell line project. So once you click on this file, so here you need not to upload any file, as this is a demo. We have already provided the demo input file. So in the input files, actually we have FASTQ files from RNA-seq data, and these files under the input files, this is how we have already uploaded our data. So which contain FASTQ files for RNA-seq from the RNA-seq data for this particular cell line. So then once you click on a start, so then you will find there are different options that are getting highlighted. As here we are just trying to visualize our sequences on a reference genome. So I'm just skipping this step, pre-processing here only. I'm just skipping this step. Then I'm just going for the next step, that is mapping on a genome. So here you will find different algorithms that can align your sequence reads on a reference genome. So I am here selecting Butai 2TG. So it will align your sequence reads on a reference genome. So then we will get an option, that is visualization. So after clicking on the visualization, so we just clicking on a OK. So the next option that is getting highlighted, that is end. So while clicking on a end, it will complete our pipeline. So once your pipeline get completed, so here you will find different files. So that is under the output files, you will find various output files under the download pipeline outputs. The other option that here you're getting, that is genome visualization, and the blue, in the blue it is getting highlighted. So you can click on this visualization and you will reach to this particular page. So here, as you can also play with the different regions. So here this finally you will just next move to this JBrowse page. So this JBrowse interface showing you alignment of different reads on a reference genome.
Dr.
So
Harpreet, can you just repeat yourself over? There are some people who are not able to catch up. So if you can just a bit more slow. Thank you.
Okay. Okay. So are you able to complete this pipeline? So let me just tell you again. Once you click on a start, then go to this under the mapping on a genome. Here you will find different algorithms of mapping. So you just click on the next option, that is Butai 2G, Butai 2G. So if you're able to this, then click on OK. And then you will find another option, that is visualization. So once you click on visualization and click on OK, then you will, the next option that is getting highlighted, that is at end of this file, it would be an end. So once you click on end, you will complete your pipeline. So on completing this pipeline, so you will find here different options. So like output files, and under the genome visualization, there is a one option that you are not getting, that is visualization. So if you click on this visualization, you will next move to the JBrowse page. I hope every one of you able to get this point. Just let me know.
So we will be addressing the details we will be going in depth in the second session that Dr. Harpreet has shown. This is just a demonstration for this free webinar. And obviously, once the program starts, there will be a dedicated session for genomics data analysis, where we will be going through in much more detail, learning from the data set and etc. So thank you, Dr. Harpreet. And now let's just go back to the presentation and try to look into the other platform.
Okay. So now let's back to the presentation. And this is how you can visualize your genome on a visualize your alignment of sequencing reads on a reference genome. So beside on under this genomics program, genomics sessions, here we will also learn about how we can perform mutational analysis using this Bioinfo platform. So in the mutational analysis, the first step that we will go for the pre-processing. Here it will remove the PCR duplicates using the PCR clean, and it will also remove the adapters that using a Trimomatic. Then the step that we will perform, that will go for the mapping alignment, where your sequences, you will align your sequence on a reference genome. Then the next step where we can compute the variant callings or the mutations. It will determine which nucleotide positions have a change from a reference genome and differentiating consistent changes from the random. Then we will go to the next step that will perform differential variant callings. So here we can compute the significance of the variance between two different groups that that are of our interest. So we can perform using different algorithms like Strelka. So then we will this platform offer you another option, that is the annotation, where you can compare your variants with the reference database to see whether the information exists or whether you find a new or rare variants. So this is we will perform this demonstration of mutation analysis during our upcoming program. To supplement with the online sessions, you will have access to a genomics section at our educational platform, where the methods and the data sets are described in much more detail. So if Vipsa, you can share the link for these courses, that would be great. So in the next session, we will look at the gene expression data, and we will try to learn how we can measure functional protein by the way of mRNA abundance and other analysis processes. Here, we will also learn about quantification of the genes and the RNA transcripts. Then we will learn how we can prepare a data for any analysis. Here we will learn about various types of data sets and their properties to understand the process of data analysis that are taken from published literature or publicly available data repositories. Then in the next session, we will learn about how we can generate gene expression data from a RNA-seq pipeline from the raw reads using using a RNA-seq pipeline at Bioinfo platform. Then our next session will be dedicated on a BioML. That is where we'll learn about the applications of machine learning in next generation transcriptomics data. So on the October 27, we will have an entire session dedicated to code omics logic playground. Here we will learn about how to do coding in R and the Python for various types of data analysis. So now let me, so at our coding omics logic platform, it has various tutorials that will help you to learn and understand the coding in the R and the Python for various kinds of data analysis. So let me take you to the code omics logic platform.
Yes, Dr. Harpreet, and if you can just quickly show us the courses that are available there and things people can do, like we don't have much time. So yeah.
Okay. So once you move to this code omics logic platform, here we will find different courses like getting started with bioinformatics, in R and Python, then introduction to the data science in both in R and Python, then genomics, metagenomics, transcriptomics. So let me just demonstrate with how we can under this course. So in the getting started with the bioinformatics R, you will find different lessons. So let me just give a simple demonstration how we can load data in R. So how you can do this in R. So when you click on this lesson, so this under every single lesson, you will find a complete details of every single step. And so it started with the first to understand the problem. So here you will get details, but what are the problems that we are going to learn under this particular lesson. So it will provide you detail. So once you complete and then you move to the next step. So the next step you will learn, the next step is about learning the code. So here you will find the code breakdown. So every single step here is explained and how you can import your data. For instance, here we just trying to import a data in a R. So in every single step, it will explain what we are doing. For like here, first we are just importing a data in R. Here we are just uploading a data file using a text, that is a text file. So you will find complete detail, but how we are doing, we are performing every kind of analysis in our R. So every single step is will explain you step by step. So once you understand the how you can write a code, so you can complete it and then a text. Then the next option that you will get, then in the next step you can try it yourself. So for instance, here we are just trying to upload a data. So like how I can import a text file in R. So let's look at how we can do this. So here with the read dot table in the R, this command, and then here it will explain that you should provide the path of your file. So I have already file you. Let me just do it by myself. So df, in the df we are storing our data that is that we are uploading. So read dot table will actually read your text file. Here you can find you can paste the path of your input file. Then you have to define so separator, that is whether your file is tab separated or a comma separated. So like here in my case this is tab separated. Then we have to just tell whether header is true or not. So if there is a header in a file, so then we can just write is true. So this is a simple step how you can upload a text file in a R. So if I run this, so it will show me that this particular file get run, and I have to how I
So,
can check
Dr.
whether my
Harpreet,
okay.
if I may interrupt, so I mean, we will be again discussing this in much more depth in the session. So maybe if you
Okay.
don't go into such details, because we will be covering this, and it's difficult
Yeah.
to explain everything in such a short time. And people are some like Farheen here is finding it difficult to understand. It's because we are doing it in a really quick session, Farheen,
Yeah.
and this is an overview. So if you can just like move on and
Okay.
go to the next section, and yeah.
Okay. So this is how you can actually run your code. And here at the Codeomics Logic platform, you will find the details, and you can write your own code, and you will learn it in detail. So once you complete, and you will find a different you can complete it, and then you can also you will ask to complete different assignments. So this is how this Codeomics Logic platform will help you to learn to write any code in R or the Python. So now I would let me pass this session to Dr. Mohit. So
Thank
over
you.
to you, Dr. Mohit.
Yeah. Thank you, Harpreet. Thank you for this wonderful presentation. And I mean, I just wanted to announce this. People who are interested in going in much more depth about R and Python, so and data science. So we have a dedicated program which will be more detailed about just data science aspects and Python and R programming. So, Bipsha, if you can just share that program details in the chat, the data
Yes,
science
Dr.
program.
Mohit, I will just be showing that after I address the registration process. So you can move on.
Perfect. Yeah, perfect. Let me just quickly, finish the session which we will be covering. So far we have discussed 5 sessions on different data types. Some of the silent features of this coding platform is you don't require to install, Python and R in your computer, but you can do that as well and do it alongside. And this is one-click run, and you will be getting points. So it is built for biologists and clinicians and students to learn. So we are trying to make it more easy, and we'll be supporting it. We'll be there all the time through different forums and stuff, which Diksha will be talking about in a while. So let me go back to the last session and tell you that what we have planned after this. So this will be on 27th October, and then. Yeah, so if you were like, these are the two logins that we have shared with you. So Diksha has already shared them in the chat, but you can take a screenshot as well. So once we are done, and once we have learned this technologies, learning about big data in life sciences and understanding the properties of such data sets and the intuitive platforms and its functionality. We will be covering all that by this time. And we will have a dedicated session on bioinformatics big data project. So in this session, we will learn to ask questions such as, what is big data research project is about? What is the research question that you're trying to ask? Can this data help in answering those questions? So these are the things, and we have a lot of participants who have gone through this entire process and have done excellent video presentations and poster presentations at the end of the program. So this so to do that, however, to understand this application and to be able to find information from this big data, it is important to learn from the analysis strategy and get experience working on this real data set. So together with our research collaborators and from top universities, we have prepared projects for you that takes you through a project-based approach where we take data sets from these high impact publications and use them for practical exercises. So in this 1 month training program, will include several components, as you have seen an overview. With this information, let me pass it to Bipsha to talk to you about the registration process and things like that. So over to you, Bipsha. Thank you, everyone.
Yeah, thank you so much, Dr. Mohit. So I will quickly show everyone some of the things that I wanted to share about who this program is for. And this program is for anybody who is starting out with bioinformatics. So you can be in high school, you can be in undergraduate or postgraduate level, as well as for faculties, biologists, and clinicians working in labs and exploring multi-omics data. So I also wanted to highlight some of these achievements that previous program participants have achieved. So here we have Anurag Kanasi. He was part of an Omics Logic program in summer, and along with Arva Bora, they were part of the winning pool team challenge area in HCL Better Health Hackathon. Then we have Urja Parekh. So Miss Urja Parekh is our research fellow. She recently completed her bioinformatics internship with us, and she also completed a project titled Differential Identification of Biomarkers between Neural Stem Cells and Glioblastoma Stem Cells, and is continuing her research in the area of neuroscience. This is Lashanda Williams. She was part of our Omics Logic program on infectious diseases, and she was recently accepted to the fall 2020 cohort of the Data Incubator Fellowship Program. So these are just an overview of what are the possibilities of doing these programs, because these programs are all research-based programs, And now coming back to the registration process. So this is the link where you can register from. Let me just post the answer in the chat. Okay. So yeah. So Krishna, I totally missed your question again. So can you please repeat it for me once more so that I can answer your questions quickly? So this is the registration link to this program. And once you go to this page, you will be able to see the different dates this program is going to be covering that already we have given a demonstration on. And each of these sessions will start at 9 a.m. CST like today, which is 7:30 p.m. IST. And fees and certification eligibility, right? So once you click on get started, you have 3 options to choose from. You have a basic option, which is $70. And then you have an intermediate and advanced option. I will just explain the differences quickly between these 3 options. So the basic option will give you access to all of the resources that were discussed today, which is the educational portal and the courses, the server Bioinfo platform, and the Genomics logic platform. What separates it from intermediate is that if someone chooses the intermediate membership option, they will be able to have a one-on-one session with a mentor. So you've already met our mentors, you will be able to go on a call. And if you have any kind of problems, any kind of issues, so you can avail that one-on-one mentor session. The advanced option is for anybody who is going to be working on a research project. So I just showed you one research project that Urja had done. So what this allows you is another 30 days extension of your membership. So if you are just a student, you can start with a basic option, Bhumika. And the certification eligibility is that you can come from any background. And most importantly, we give out 2 types of certificates. I will just show you here for an example. Let me just go here. So you see here, this is a certificate for Mikhail. So he has a certificate of excellence. So how is it calculated? It is calculated right on these points that you are seeing here. So you earn these points by doing the different courses that we just demonstrated, like the introduction to bioinformatics. So you can actually go and do the different courses, and you see that with each course you get some points. Each pipeline you run on the server will add points to this, and the more courses you do, the more points you will earn. So if you have over 10,000 points, so that is what we have kept the limit. So 10,000 points are certificate of excellence, and over 2,000 points for certificate of completion or participation. So if you do not do anything, then we do not usually offer a certificate for everyone. And if you see, Mikhail here has been part of few different programs. So he has 2 certificates here. And also you can see his independent project. If you would just like to take a look at his profile, you can just take a look. So again, this is the registration link. You go down here, you enter your details, and you click on any of these options. So somebody asked me, Sabila asked, like, what is the better option for MPhil? So it really depends on where your level of bioinformatics is, as Ilya has already said. So it will be depending on you. If you're not able to understand something, or you think that you would need more support than what is already provided in the program. So how do we provide support in a program? That is exactly what we do through this page. So when you're enrolled in the program, everyone will have access to this page. And this is called an organization page. And here is a forum. So this is the forum where we used to discuss, where we work together as a team, and we communicate with you. So you can directly communicate with mentors, and this does not require any other membership other than a basic membership. The intermediate will be like a one-on-one Zoom call or a one-on-one Skype call. So that would be more of a one person and one mentor situation. I hope that is the difference you're able to understand. But this forum is open to everybody who is part of this program. And we will be adding more topics, we'll be adding more things which you will require during the course and carrying out the entire communication process here. And here are the courses that you will have access to, all for free. So you would not have to pay for these courses. So Introduction to Bioinformatics and Introduction to Genomics are free, but from Genomics 1 or the entire transcriptomics series or even metagenomics is not free. They each cost $10. So that is inclusive with the cost that you're paying for the program. Plus you also have access to projects. So you see, we have a few projects where you will be able to learn from. And these are also included. No, you cannot get a certificate without enrolling in the program. Each program will have different certification and they will have different levels. So this program we recommend for anybody who's starting out with bioinformatics and you're really wanting to get familiar with these concepts. So you can join this.
Yeah,
Yes.
Disha, and you can tell that each of these courses and projects, you will get an individual certification for completing those. So
Exactly.
if you
Yeah.
complete Introduction to Bioinformatics, which is a free course, you will get a certificate. If you complete the Introduction to Genomics, you will get a certificate. But for program certification, you have to register and take the entire program with us, where there will be live sessions and all the things that Disha has talked about. And we'll be discussing about your
Right.
progress and all this.
Yes. Thank you so much, Dr. Mohit. And also, we can also track all of your progress here as a group and also individually. So this is also available if you are a faculty and you want to see that if your students are performing well, or you want to see the entire group and how their performance is in a program, we are able to see that via this site. Everything is tracked, so you can see whatever you're doing. On Codomics Logic, it appears here. On server, it also appears because there is no server currently run. And also on the Edu site. So this is the entire thing that the program offers to you. And along with that comes certification. So I hope that this was much more clear to you. If you have any questions about this process of registration, please let me know. You can always email me marketing@pine.bio. And I have already received all of your emails. If anybody did not leave your email and you would still want to receive details about these programs, please let us know. There are also a few blogs that you can read here. These blogs are for free on our Edu profile. You can see what are the different participants saying about the courses. You can read about this one by Mikhail. He shared the 5 reasons why he recommends Omics Logic training program. Okay, thank you so much for sharing these details. And I also wanted to share more details about the groups where you can find more information about any of these upcoming courses. We have these 2 groups on LinkedIn and on Facebook.
And also job opportunities. So we post about bioinformatics related job opportunities and updates from our participants on who are doing well. And if they, we have people who are joining from different companies and different academic institutions. So that's a community that you can leverage.
Exactly. So you do not only do a course on your own, you also interact with this entire community of students, faculty, bioinformaticians, and other industry specialists. So that is a great opportunity. And for others who would be interested, we do have some programs. Oh, I'm sorry, Doa. I don't know why you cannot hear my voice. Shomo, for a free webinar, we usually do not offer a certificate. You can actually go and do the Introduction to Bioinformatics course because that has a certificate. So you can go and do the course and you can earn a certificate and download it and share it in LinkedIn. So I just wanted to point out 2 more programs that will be starting later. So this is also starting on October 12th, and it's on data science. This is for anybody who wants to learn more about data science and wants to particularly work with biomedical data using R and Python. If you're interested, please take a look. And we also have one for metagenomics. I did notice that there were a lot of people who are working in this domain as well. So this is starting on 17th November. So these 2 courses are also available for anybody else who is a bit more advanced and would like to explore these other 2 courses. So if you have any questions right now, we are open to taking questions. Was there any question that we missed?
No, Bisha, I think we have addressed the questions, and obviously you can reach out to us with all the different platforms that we have shared, and you have already logged in into the educational platform, so you can always ask us there as well. So yeah, we are repeating these courses every year, and almost like every quarterly we are doing it. So if you are interested in some course that you wanted to take in next year, we will be most probably. We also have transcriptomics course, which we will be starting in December, and infectious diseases that we completed in summers, and we will be again doing infectious diseases. But this is an introductory course that actually is going to help you to do those advanced courses, because you need to get familiarized with the data and then the application of the data. So yeah.
So for everyone who has been asking about, introductory courses, or you're just starting out like Bhagyashri, Sabila, and others, I would recommend the Omics Logic Introduction to Bioinformatics, because it's really an introductory course. So we will start at the beginner level and help you learn about bioinformatics. If there are no more questions, so I thank you. Yes, the deadline, as you see already, it's October 13th, the course is starting. So it's just next week. So you can just sign up within these 2 days.
Yes, to answer the questions, collaborations for papers. Let me.
For Christian, yes, Dr. Mohit.
Let me answer that question from CA about the collaborations for papers. So publication takes a while. So we start from the very beginning, understanding the technology, then we progress to the research question, and then the analysis there. So the research project is also divided into several aspects and several different landmarks to be able to do that. So once we reach there are several opportunities, like we said, that we have a huge group of bioinformaticians associated with us, and not only bioinformaticians, scientists from every aspect of biomedical research. So there is an opportunity to collaborate at a later stage once you have defined your project and you know what you're looking to. And if you have a research project, you can obviously do research and collaborate with us as well.
Yeah, thank you so much, Dr. Mohit. Yes, Dr. Mohit, if you also answer this.
Yes, I can see each other question. So Irmac, yes, I think the if you see like on the screen, it's Omics Logic. So we focus on the logic. If you see, logic is bold. So we focus on the logic. So once you learn the logic, once you understand that you can do it anywhere, it does not matter about the platform. You can do it on your own computer. You can do it on some other platform. So it's important to understand the logic and then go about and implement that logic into different research problems.
Yeah, thank you so much, Dr. Mohit. So I thank everybody for joining today. I will reach out to everybody who has shared their email IDs and share the video link and the details of the registration process again. Thank you, everyone.